Hello everyone. In this video lecture, we will discuss about the details of a prokaryotic cell. The diagram is there in front of you about a prokaryotic cell, that is a bacterial cell. Now, prokaryotic cells are basically represented by bacteria, right? They are represented by the mycoplasmas and they're represented by the PPLO, that is a pleuropneumonia-like organism. Now, many of the time the question arises in mind that why they are called as a pleuropneumonia-like organism. Basically, the mycoplasmas, they do not have the cell wall, and as they do not have the cell wall, the shape of the cells may change. So they are present in many different forms. So that's why they are called as pleuro, and they cause pneumonia when they when they enter inside the host organism, inside the human body. So that's why they are called as a pleuropneumonia like organisms. So bacteria, mycoplasma and PPLO, they represent the prokaryotic cells. Okay. Nextly, they are basically very smaller in size, and when they multiply, they take a very less time to multiply as compared to the eukaryotic cells. Okay, in the bacterial cell it may multiply within 15 to 20 minutes. That is a generation time of a bacterial cell. So that is how the bacterial cells multiply within such a short period of time. They may vary greatly in their shape and sizes. When we talk about the bacterial shapes, okay, there are some bacterial shapes which we should remember. These are the basic shapes. Some bacterial cells are present in the form of rods, which are called as the bacilli. Okay, the bacteria is called as bacillus. Then some bacteria may be formed in a spherical shape. These are called as the cocci, or it is called as the coccus. Some bacteria may be present in a spiral form, which is called as the spirilla. Okay, and or the spirillum also they might be called as, and some are present in the form of a comma. Okay, so these are called as a vibrio. Vibrio cholerae is the best example of this comma shaped type of the bacteria. So this is the vibrio type of a shape. So there is bacilli, there is cocci, there is spirillum, and then there is a vibrio. The organization, when we talk about of a prokaryotic cell, the organization, it is very simple and it exhibit wide shapes, wide varieties of shapes and wide varieties of functions. They have a cell wall. Okay, they have a plasma membrane. Yes, all cells they have a plasma membrane, but outside of the plasma membrane they have one more structure, that is the cell wall. And the cell wall in the prokaryotes, it is made up of peptidoglycan. Based on the composition of this peptidoglycan and the teichoic acid that is present and the lipids that are present, okay, this bacteria can be divided as gram positive bacteria and they can be called as gram negative bacteria. There is a staining procedure, okay, in when we have to observe the microscopes under the when we have to observe the bacterial cells under the microscope, there is a universal staining procedure which is called as gram staining, and this gram staining is carried out same for all the bacterial cells. But it happens to be that some bacterial cells they obtain purple color and they appear to be violet or purple color under the microscope, and some bacteria they may occur pinkish in color under the microscope. So the bacterial cell which appears to be pinkish, they are called as a gram negative bacteria, and the bacteria which appear to be purple to be violet in color, let us take violet, they are called as a gram positive bacteria. So that is a gram staining procedure. I will be making a separate video on the gram staining procedure because it's a big procedure and it is not given in our syllabus what is the procedure is actually about. But to understand in detail about the gram staining procedure, I will be making a separate video for it. If you like, you can watch. That's up to you. So moving on to the next point, that when we talk about the cell membrane, okay, so the cell wall is present in the prokaryotic cells that we have talked. The cell membrane is present in all the cells, but the mycoplasmas, they do not have the cell wall. Okay, bacteria, yes, they have the presence of a cell wall. Coming to the point of nucleus, Okay, the nucleus is absent, we have discussed in the previous video. So what do they have? Their genetic material, their DNA is coiled around. Okay, it is tightly coiled around. Now the DNA is a negatively charged molecule, so if it has so if it has to be coiled around, it requires some attraction so that it can be coiled around to it. So there are some positively charged proteins, and these positively charged proteins are called as the histone like proteins. Okay, remember that histone proteins are present in the eukaryotes, but the proteins which are positively charged, they are called as the histone like proteins that are present in the bacteria, and they help to coil a long DNA molecule. The DNA molecule, if we extract from the bacterial cell and if we just make it linear, okay, अगर हम इसको straight करेंगे तो it will be around 1.2 meters long. Okay, so this 1.2 meter long DNA is coiled and it is fit inside such a microscopic cell, so that all miracle is done by the histone-like proteins. Now, why they are positively charged? Why these proteins are positively charged? Because we know that the proteins are long chain of amino acids, right? There are 20 different types of amino acids, and there is a structure of amino acid, a basic structure that I will draw, that it has a carboxylic end and it has an amino group, and there is a R chain. So if this R chain is basic, the charge that is going to be positive, and because of these positively charged amino acid, the proteins they become positively charged, and the DNA is negatively charged because of the presence of phosphate, right? So that's why the positive and the negative are going to attract, and it is how the packaging of the DNA takes place with the help of the histone like proteins. Now when this packaged DNA is present, it will be called as the nucleoid. It is called as a nucleoid. So this is a chromosomal DNA. Leaving this DNA apart, there are some small circular DNA which are double-stranded DNA molecule. Those are also present in a bacterial cytoplasm. Now these circular DNA are called as, let me write here, they are called as the plasmids. Okay, plasmids, they are extra chromosomal. Extra chromosomal, that is, they are not a part of the chromosome of the bacteria, but they are extra chromosomal DNA molecules which can replicate on themselves, which can make their own copies. Okay, they can go on replicating if required. So these plasmids, this plasmid DNA have their own specific genes. In some bacteria, it is seen that these genes are responsible for giving the bacteria a special type of a feature, that is antibiotic resistance. Antibiotic resistance. So the genes for antibiotic resistance are present on the plasmids. What is an antibiotic resistant feature? When we talk about antibiotic, these are basically the chemicals which are produced by the living organisms which kill or stop or inhibit the other living organism. It was accidentally formed by Alexander Fleming when he was working on Streptococcus, and when he was working on the Streptococcus on the Petri plate, as he kept the Petri plates open, a fungus started growing on that Petri plate, which was later on called to be penicillium, Penicillium notatum, and it was found that when the Penicillium notatum was growing, it released some chemicals, and now it is came to know as the penicillin. Later on, it was come to known to be penicillin, which was the first antibiotic that was discovered by Alexander Fleming, and that was used widely to treat the World War I patients, and also it was a boom after that. So it was an accidental discovery, but now we know them as antibiotics. So some bacteria, they are not resistant to all the antibiotics. Specific type of antibiotics they might be resistant to. So this is a small feature of the plasmid. Plasmids are also widely used in the field of biotechnology. Okay, there is a whole complete chapter on in the second year in 12th standard on biotechnology and the use of the plasmids. So just what we have to remember is that these plasmids are used so that a different type of a gene can be inserted into the plasmid, and this plasmid is then inserted into a bacterial cell. So when the bacterial cell multiplies, the plasmid also multiply, and the plasmid has a specific gene, okay, which is required for the human welfare to obtain a specific protein. So that gene also expresses itself and the protein is produced with the help of these plasmids. So it is a long information about the plasmids. We will have a separate video for the plasmids from the chapter related to biotechnology for 12th chapter. But just remember that plasmid is widely used in the field of biotechnology, especially in rDNA technology, that is recombinant DNA technology. Okay, so this is all about the plasmids. When we talk about the ribosomes over here in the bacterial cell, ribosomes, we have to remember that ribosomes are present in eukaryotic cell as well as they are present in the prokaryotic cells. Ribosomes are very tiny structures, and in the eukaryotic cell, they are basically free in the cytoplasm, or they are present on the surface of endoplasmic reticulum. Okay, and such endoplasmic reticulum which has ribosomes on it, it is called as a rough endoplasmic reticulum. We will see why it is called as rough later on when we start about the endoplasmic reticulum. Now, when we talk about ribosomes, the ribosomes has two subunits. One is called as a large subunit. Sorry. One subunit is called as a small subunit, and the another subunit is called as a large subunit. Okay. The small subunit in a prokaryotic cell is considered to be 30S. This S is basically the Swedberg unit. It is a sedimentation coefficient, so it is 30S. So alone when it is centrifuged, it sediments, okay, and the sedimentation coefficient which is found to be 30S. When we talk about the large subunit is found to have 50S sedimentation coefficient. That is the Swedberg unit is 50S. That is when alone the large subunit is centrifuged, it sediments at the bottom of that centrifuge tube, okay, at the 50S sedimentation coefficient. But together when they are centrifuged, their sedimentation coefficient is found to be 70S. Okay, so we don't have to calculate this according to the usual mathematics that 50 plus 70 is equals to 80. Here it is about the sedimentation coefficient that has to be considered. Okay, so this S, it tells us about the Swedberg unit. So prokaryotes have 70S subunit, 70S ribosome, and this 70S ribosome is made up of two subunits. Small is 30S subunit and large is 50S subunit. In the same way, the mitochondria of a eukaryotic cell, mitochondria also has 70S ribosomes, okay, of their own, and chloroplast also have the ribosomes which are 70S. Okay, so this is a common thing between the organelles which are present in the eukaryotic cell. Even though they are present in the eukaryotic cell, they have these kind of ribosomes. But when we talk about the eukaryotic ribosomes are 80S. Okay, they are made up of two components, 40S and 60S, right? And together they make up to 80S. Right, so this is about the eukaryotic ribosomes which are present in the cytoplasm, and they might be present on the surface of endoplasmic reticulum, which is called as a rough endoplasmic reticulum. So this is about uh, the same organelles which are present in the eukaryotic cell as well as in the prokaryotic cell. Along with these ribosomes, they have some structures which are called as the inclusion bodies in their cytoplasm. Now, what are these inclusion bodies? Inclusion bodies are very tiny structures present in the cytoplasm. These inclusion bodies can be gas vacuoles. Okay, gas vacuoles are found to be in purple and blue-green bacteria and green photosynthetic bacteria, right? So these are the gas vacuoles where maybe the different types of gases are stored. Okay, so they might be helping these bacteria to float on the water surface. So that is about the gas vacuoles. There are inorganic inclusion bodies which are called as the volutine granules. These are small structures, granules are present. Then also it is found that some bacteria, they have magnetosomes, and these magnetosomes help the bacteria to align themselves according to the north and the south direction. Okay, so those are the magnetosomes. They also have some granule which store the sulfur, which are called as the sulfur granules. Some of them, they have PHB granules which are called as poly beta hydroxy sorry, butyrate granules. Okay, so these are some of the granules that are present. Then cyanophacian granules. The cyanobacteria has cyanophacian granule inside which the cyanophacian starch is stored. Okay, they also have glycogen granules inside which the glycogen is stored. Right, so these are all about the inclusion bodies, the ribosomes that we have talked about. And now, following this point, let us talk one more thing in the prokaryotic cell, that is the cell envelope. Right, all the cells, the living cells, they have the plasma membrane, but outside the plasma membrane we just studied that the bacteria has cell wall. Okay, this cell wall is supposed to be made up of peptidoglycan. Now why it is called as peptidoglycan? It is a part protein and it is a part carbohydrate chain, so it is called as peptidoglycan. And N acetyl glucosamine and N acetyl muramic acid. These are the two components of the peptidoglycans which make up the cell wall of the bacteria. Okay, and as we discussed about the gram positive and gram negative bacteria, there are some more things that we have to focus on, that is teichoic acid and the lipid membrane, that we will discuss in a separate video. So this is the composition of the peptidoglycan, this is the composition of the cell wall. On the sorry, in some bacteria it is also found that on the outer side of the cell wall there is one more membranous covering that is present. Okay, and so it is tightly bound to the cell wall. This, the outermost, is called as the glycocalyx. Okay, so the glycocalyx, the cell wall, okay, and the plasma membrane, all these three together, it is called as a cell envelope. When the glycocalyx is present in a loose form, or when it is a loose layer of the glycocalyx, this will be called as a slimy layer or a slime layer. But when it is present in tough and in a rigid form, it is called as a capsule. Okay, so this is a slimy layer or slime layer or a capsule. Okay, the glycocalyx that can be called as and this capsule or this slime layer basically determines, or the cell envelope determines the shape of the cell, we can say. It provides a strong structure, and so that the bacterium should not burst. Okay, the bacterium should not burst or the bacterium should not collapse under unfavorable conditions. Right, so this is all about the cell envelope. Coming to the next point, the bacteria has one more structure which is called as a mesosome. What is this mesosome? The plasma membrane of the bacteria has some infoldings. Okay, these infoldings are present at some particular regions which are called as the mesosomes. These mesosomes play a very important role as they are involved. They will help the chromosomal DNA during the time of DNA replication. These mesosomes also, they form the vesicles, the tubules, and the lamella inside the bacterial cell in the cytoplasm. They also help in the DNA distribution during the time of the daughter cell formation. Okay, that is one thing. They also help in respiration, because the electron system or the electron transport chain, where the electrons has to be transported, the ATP has to be generated, so that all takes place with the help of the mesosomes, the respiration. Then the mesosomes also helps in secretion. Okay, secretion processes. They also help in increase the surface area of the cell, because the more the infoldings, the more will be the surface area of a cell. Okay, so that is how the mesosomes helps in increasing the surface area. In some prokaryotes, like some cyanobacteria, which carries out photosynthesis, it is found that these mesosomes they extend and they form a structure which is called as a chromatophores, and inside these chromatophores the pigments for photosynthesis are stored, right? So it contains the pigments. And now the last point in the prokaryotic cell, let us go to the diagram. So we have the structure in the diagram which will give us an idea. Some bacteria are found to be motile, that is they can move from one place to another place, and some bacteria are found to be non-motile in nature, that is they cannot move on their own. Okay, this motility is caused in those motile bacteria because of this thread like structure which is called as a flagellum. Okay, this flagellum is made up of a protein which is called as a flagellin protein, which helps the bacteria to move forward or backward direction. This flagella has three parts, okay, in the cell wall. They are adhered to the cell wall. It is called as the basal body, which is exactly at the position of the cell wall. They have a hook region. So I will write it over here. They have a basal body. They have a hook region, and from this hook there is a filament. That is the tail region that is present. Okay, so they are going to help for locomotion. What are another structure? Okay, so another structure that is present are called as a pili. Okay, it is found to be that pili help during sexual reproduction in the bacteria. Okay, now what is sexual reproduction in bacteria? Let us understand. Pili, if the pili factor is present on any bacterial cell, these bacteria are called as F plus. If this is a bacteria and if it has a pili, it is called as an F plus. Okay, so this is a fertility factor that is present, and if this is a bacteria, it does not have a pili, it will be called as F minus, that is the fertility factor is absent, and this pili forms a conjugation tube. This is called as a conjugation tube between these two bacteria, and the replicated DNA can be exchanged from one cell to another cell. Okay, so this is called as a mating, or this is called as a sexual reproduction in bacteria where the pili may be involved. So this is about the pili, and the another structure that is present is called as the fimbriae. Now what are these fimbriae? If it is a bacterial cell, okay, the bristle-like structures are present on the surface of the bacterial cell which help the bacteria to adhere to some structure when it enters inside, maybe inside a host body, so that it can adhere to a structure and it can help the bacteria to multiply and to cause the infection. So this is about the fimbriae. Okay, maybe if they are present in water to stick to the rocks, okay, in the streams, okay, or to the host tissues if they are inside the host organism, these fimbriae they help the bacterial cell to adhere itself, to catch to some specific regions. So this is all about the prokaryotic cells. In the next video lecture, we will discuss about the eukaryotic cells. I hope you have understood. If I hope you have understood this. If you have any doubts, queries, you can contact me in the comment section. Thank you so much for watching the video.